A dendritic cell, a class of phagocyte which has been created through clonal selection, is drawn to an area of infection by the release of chemokines from damaged cells bringing into contact with the pathogen. The dendritic cell engulfs the pathogen, lyses it so that only short polypeptide chains are remaining. These polypeptides attach to proteins forming MHC2 complexes which migrate to the cell membrane. The dendritic cell can then be labeled as an antigen-presenting cell. A CD4 positive helper T cell whose T cell antigen receptor has a variable region which is compatible with the dendritic cell's specific MHC2 receptor. This allows the helper T cell to become activated. The activated helper T cell then proliferates into thousands of memory helper T cells as well as effector helper T cells. The effector helper T cells release cytokines which sounds an alarm, alerting the rest of the immune system of the infection. A cell has been infected by a virus and begins producing faulty proteins. MHC1 within the cell attach to these faulty proteins then migrate to the cell membrane. A CD8 positive cytotoxic T cell whose T cell antigen receptor has a variable region which is compatible with the infected cell, attaches to the infected cell. This allows the cytotoxic T cell to become active. The cytotoxic T cell then proliferates into thousands of cytotoxic memory and effector T cells. An effector cytotoxic T cell attaches to the MHC1 complex. After the cytotoxic T cell releases perforin, which bores pores into the infected cell. After the pores have been created, the cytotoxic T cell releases granzymes. The granzymes trigger apoptosis, forcing the cell to kill itself. The cytotoxic T cell is released after.